Our next speaker is Dr. Saima Khalid, research scientist from USA. The topic of our discussion is role of microbial communication in well-being of human health and agriculture. Over to you, Dr. Saima.
In the name of Allah, the most beneficent, the most merciful. I'm Dr. Saima Khalid, and I will be presenting role of microbial communication in the well-being of human health and agriculture. This information is taken from a recently published review about microbial communication. Microorganisms reside together into different environmental communities, and just because of living together, they interact with each other and they have certain kind of communication among them due to close physical interaction. Just like higher organisms, since microorganisms also live together, so they have certain kind of communication with each other in the form of social networking, and the field of studying that kind of communication between microorganisms is known as socio-microbiology. Communication is basically a message that is given by certain microorganisms that live together, and it is being perceived, processed, and then responded to through different phenotypic and genotypic changes. It means that when microorganisms live together, they give certain information about the change of the environment in the form of a message or a communication signal that is given to other microorganisms, and they show the message to be received in the form of certain transformation into their metabolic as well as genotypic changes. Microorganisms have this kind of microbial communication by using different secondary metabolites. They are equipped with diverse small signaling molecules or secondary metabolites, and those secondary metabolites are of different kinds. For instance, antibiotics. All of us know that antibiotics are basically produced to kill or inhibit the growth of partner microorganisms. Or in addition, they also produced volatile organic compounds that are chemically diverse, small lipophilic metabolites. Quorum sensor molecules are also known as autoinducers in bacteria. They are synthesized in a chemical gradient form, and they are detected and received by microbes, and they result in developmental shifts that are appropriate to the environment. Although these all secondary metabolites are produced by microorganisms to perform different functions for them, but at minimal concentrations, all these secondary metabolites also have the ability to act as signaling molecules. And these signaling molecules, they have the ability to induce certain changes into the co-partners like estin production, motility, as well as biofilm formation. Bacterial fungal communication is a hot research topic nowadays. Bacteria and fungi reside together into different environmental conditions, ranging from the soil samples to the communities on host and within the host, and as well as on the outside of the host body. Since they share a same habitat, so they result into different interactions, and those interactions are basically carried out due to communication between these two kingdoms, and this communication results into different transformational changes into bacteria as well as into fungi. For instance, in this example, we can see that when Aspergillus flavus, that is a fungus, when it interacts with the bacteria, Ralstonia solanacerium, so in fungus, the IMQ-K gene cluster, that is amisoquine gene cluster, its expression is reduced. As a result of this, interaction and germination is also reduced. On the other hand, in bacteria, the growth rate is reduced. It means that when these two microorganisms come together, they result into certain transformational changes that are represented in the form of certain developmental changes that we can see. How these small signaling molecules are involved into drug discovery. For instance, in this example, we can see that Aspergillus fumigatus, when it interacts with Streptomyces leeuwenhoekii, and then it leads to inhibition of certain antibiotics, for example, enzymycin as well as macrolactin, into the bacteria. And in addition, there are certain metabolites, for instance, lysopeptide, that are increased into bacteria. And however, in the fungus, there are certain secondary metabolites, their production is reduced. For instance, in addition, there are certain antimicrobial metabolites such as glutarate D and serotonin G. So their production is induced into fungus due to their interaction with bacterium. It means that, as I said before several times, whenever bacteria and fungi they interact with each other, they induce production of certain metabolites, or sometimes they inhibit the production of certain metabolites. It means that their interaction, first of all, affect their genotype on the like genetic basis, and then that genotype ultimately results into different metabolite production or the phenotypic changes into the bacteria as well as in fungi. Now we will see that how these small signaling molecules affect human host in disease and health. Candida albicans and Pseudomonas aeruginosa both are human pathogens, and when they interact with each other, so the Pseudomonas aeruginosa, that is the bacteria that induce fungi in such a way that if morphological transition, that is yeast mold shift, is basically reduced, and that is the main pathogenic ability of the fungus. And also its QSM production, farnesol, is reduced, and it means that its pathogenicity is reduced. On the other hand, farnesol also reduces the production of PQS, that is Pseudomonas guanolone signal, into the bacteria, and as a result of which the pathogenicity of the bacteria is reduced. It means that whenever these two microorganisms interact with each other, they try to reduce each other's pathogenicity so that they can win the competition and they can enhance their pathogenicity on the other hand. There are certain other small signaling molecules. They also affect plant host in agriculture. For instance, Verticillium longisporum, when this fungus is grown with um, um, Penibacillus polymixa, that is a bacteria. So in fungus, the volatile organic compound duranol production is increased, as well as its metabolic activity is reduced. On the other hand, in the bacteria, as a result of this interaction, metabolic activity is increased. Certain VOC production is also increased. However, there are certain other VOC production, that is butanol, its production is reduced, or its activity is reduced. So again, these two microorganisms are interacting with each other, as a result of which genotypic changes take place, and that genotypic changes ultimately result into the phenotypic changes into bacteria as well as into the fungi. Conclusions of this microbial communication is that initially these small secondary metabolites of microorganisms were considered as microbial wastes, but now we do know that they are just they are used as very significant molecules, as communication molecules into microbial communication. They impact agriculture, as we just saw an example. So their impact in agriculture, especially into increasing the plant crop like production, we can exploit that into improving the crop production, and also to devise new biocontrol strategies. And we all know that there is a great dilemma going on due to microbial resistance to the available antibiotics. So bacterial fungal interactions help us to just discover certain novel antimicrobial agents that can be used as new antibiotics to replace the old ones against which microorganisms have got resistance. And then, as I said before, this small signaling molecules can be like produced more as a result of microbial interaction, like bacterial fungal interaction, that can also result into formation of new antibiotics. And then, as we just saw, and as an example, that in human host pathogens, like when they interact, they produce certain kind of metabolites. So by studying about those metabolites, we can also study about the mechanism of disease, and then studying about the mechanism of disease can also help us to find about certain strategies to get rid of those diseases. At the end, I would like to thank Nancy Keller from University of Wisconsin-Madison to help me write this review. In addition, I would also like to thank my advisor of PhD, Dr. Mohammed Imran, and my other colleagues for their sincere um, and sincere wishes, as well as support to help me throughout my PhD as well as later on in my career. Thank you very much.